convinced the park that this was an interesting question, an interesting enough question to ask, and they permitted us to sample Old Faithful. Now, Old Faithful is the reason they let us do it was one, you're close enough to infrastructure so that if something did happen, you could get help there fast enough.
Hey there, and welcome back to the Voices of Greater Yellowstone podcast, where we share the stories and science of the Greater Yellowstone Ecosystem. I'm your host, Emmy Reed. The Greater Yellowstone Ecosystem is known for a lot of big things. Charismatic megafauna like bears and bison, expansive forests as far as the eye can see, and mountain ranges that seem to touch the sky are all found in this unique region. However, did you know that the Greater Yellowstone Ecosystem is also home to some of the most fascinating microbes found on the planet? Enter thermophiles, microscopic creatures found in Yellowstone's geothermal regions that thrive in scalding hot temperatures. The name thermophile comes from the Greek word thermē, meaning heat, and the Greek word philos, meaning love or an affinity for something. They're not often the stars of the show when it comes to visiting the park, but these thermophiles are at the center of some of the most interesting stories and scientific discoveries in a national park. After listening to this conversation, we hope that even the greatest Yellowstone experts can head into the park armed with a new subject to explore. Joining us on the podcast today is Dr. Eric Boyd, a professor of environmental microbiology at Montana State University. For the past 20 years, Dr. Boyd has studied the microbiology and geochemistry of the thermal features in Yellowstone National Park to better understand the processes that support diverse microbial ecosystems in high-temperature environments. He's also authored over 190 scientific articles on extremophilic microorganisms, which live in environments with conditions approaching or stretching the limits of what known life can adapt to, such as temperature, pressure, salinity, and pH level. If you want to learn more about the incredible thermophiles found in Yellowstone, Dr. Boyd is your guy. We chatted for quite a while, so we've split this episode into two parts. In this episode, part 1, we'll take a deep dive into what exactly a thermophile is, how thermophiles are able to thrive in crazy hot conditions, and how Dr. Boyd collects thermophiles in the park, including some interesting methods for Old Faithful, and much more. I can't wait for you to learn all about this hot topic, so let's hop into the episode. Thank you so much for joining us today, Eric. We're so excited to talk about thermophiles with you, but to get us started, what exactly is a thermophile?
Well, it's a good question, and what we're really talking about are microorganisms, single-celled microorganisms, typically, that can grow at temperatures that range from something on the order of 41 degrees centigrade to 122 degrees centigrade. We're here in the United States. We don't tend to use centigrade, so that's roughly 106 degrees Fahrenheit to something around 250 degrees Fahrenheit. So temperatures that are well above those that make us comfortable. But these are organisms that belong to microbial domains, so typically bacteria and archaea, although there are some thermophilic eukaryotes. But again, organisms that tend to be smaller than what you can see with your naked eye. You need a microscope to see them.
And so these organisms then exist a little lower than boiling point or more than boiling point. Boiling point's about 212, but what is it, I guess, at higher elevation like Yellowstone?
Yeah, so the boiling point is about 93 degrees centigrade in Yellowstone at that elevation, right? So lower pressure, so water boils at a lower temperature. And you might ask, okay, well, what about an organism that lives at 122 degrees centigrade? Well, that's at the bottom of an ocean, right, at a hydrothermal vent, where you have higher pressure, and therefore the boiling point is higher than what it would normally be at sea level, 100 degrees.
So thermophiles seem like they should exist on an alien planet, but here they are on Earth. Can you tell me what is known about their origins?
Yeah, so thermophiles are an interesting group of organisms, and I've kind of built my career around studying them. One of the reasons people are so interested in thermophiles is because of the question of the origin of life. So if you look back in evolutionary time using what we call phylogenetic or evolutionary approaches, what you see is those organisms that branch closest to the base of the tree closest to the base of all extant life, meaning that they diverged really early, all have this thermophilic character. And so the bacteria, the early evolving bacteria, the early evolving archaea, the early evolving eukaryotes have this character of wanting or liking to grow at high temperature. And so if you use kind of the simplest explanation for that, that would imply that the ancestor of all life was a thermophile, such as those kind of organisms that you find living in the hot springs in Yellowstone. And so there's a lot of interest in understanding what those organisms can do, what kind of habitats they prefer, to try to understand what kind of conditions led to the origin of life on Earth. And this will come kind of loop back to the planetary or alien type context of your question. Um, that's why we study these organisms, to identify such locations on other planets that might have also supported the origin and evolution of life.
Is it possible to have any fossil records of early thermophiles? I mean, I know you need a microscope to see them, or is there any way to assess the contents of the rock to be like, oh, okay, maybe this was once a home to a thermophile, maybe a billion years ago?
Yeah, that's a great question. There's the earliest, some of the earliest evidence of life that we have on the planet is actually in the form of rocks. And not in the same way that you think of a fossilized dinosaur, for example. But we can look at the chemical and more specifically the isotopic signature. So different elements have different isotopes. And biology processes these isotopes differently when it metabolizes those elements. And it imparts an isotopic or a chemical signature that can be recorded in rocks. And it turns out there are some ancient rocks, 3.4 billion year old rocks from an outcrop in Australia that turns out to have been an ancient volcanic hydrothermal field. So, again, supporting the fact that life on Earth existed at 3.4 billion years ago and it had already learned how to live in a hydrothermal environment.
That's so cool. Um, how can these thermophiles exist in such conditions?
So, it wouldn't take long for you to feel uncomfortable at the temperatures that archaea and some of these thermophilic bacteria live. Um, you might have spilt boiling water on your hand or something like this when you were cooking in the kitchen or something like this. Well, so it makes us very uncomfortable. It makes other animals very uncomfortable. These organisms thrive at these temperatures. And the way they're able to do that are through evolutionary adaptations. So they make membranes, the outer components of their cells, their membranes or lipids that are different than the lipids that mesophiles or organisms that live at kind of mesophilic temperatures make. So, for example, if you were to take, and you do this all the time, if you melt butter, you heat it up. It was once solid. You heat it up and it melts.
Hmm.
Well, you do the same thing with coconut oil. And different fats or lipids have different melting temperatures, some higher, some lower. And all these archaean bacteria have done is evolve different lipid structures that have different melting points. So their lipids don't are effectively rigid, much more rigid at higher temperatures, but they're fluid, right? So they don't melt until you get to a much higher temperature, if that makes sense.
Yes.
They make proteins that themselves are more rigid and have different compositions of, for example, certain amino acids that themselves are more rigid. Okay, so when you raise the temperature, those proteins don't denature. So, for example, an egg, if you cook an egg, what you're doing is you're raising the temperature and you're denaturing those proteins. Well, if you did that to the proteins of an archaean, they wouldn't denature until you hit something like 50, 60 degrees C, depending on what that organism has optimized itself to grow at. They also make proteins that keep their DNA in the correct conformation, so on and so forth. So at every single stage of biology or biochemistry that you've learned about, typically derived from studies of things like E. coli or salmonella, more ordinary organisms. These organisms have tweaked their metabolisms. They've tweaked their protein biochemistry, their lipid biochemistry, so that it can function at high temperature.
Okay. Yeah, I'll never be able to boil an egg the same again without thinking about our thermophiles. So in Yellowstone, we've got a lot of different types. Can you tell me a bit about how many species of thermophiles have been discovered in Yellowstone's thermal features?
Another really good question. So, what we can grow, it turns out, so I'm a microbiologist, and I can readily admit this, that we are very, very not good at growing organisms from natural environments. So, on average, depending on the environment, we can cultivate maybe 1-2% of the organisms that we know are there. And we know they're there because we can use molecular approaches. So we can actually go to the environment and sequence the DNA to know who is living in those environments. And when we do that, we know that a small fraction of those organisms are in culture. And so I tell people when I take them into the park, because so many people go into the park to see the bison, the bears, the antelope, so on and so forth. And I tell them that all of that macrobiological diversity that you're so mesmerized with, I can take a single eraser sized sample of a hot spring, a single hot spring in Yellowstone, and I can sequence it and I can completely dwarf the biodiversity of the microbes that live there by the microbes in a single eraser sized subsample of material from a hot spring. And what's really magnificent about that is that's one tiny eraser sized sample from a single hot spring. And there's over 10,000 different hot springs in all of Yellowstone. So the amount of biodiversity in Yellowstone is immense. It's almost impossible to calculate without actually going to every single one of those 10,000 hot springs and characterizing them. But there are thousands of microbial thermophilic species in the hot springs in Yellowstone that we're only now starting to realize exist. And again, it's through this application of molecular techniques to these samples.
Right. So when were thermophiles discovered in Yellowstone National Park? Because yeah, you can look out your window. All right, there's some bison over there. But um did people think like, "Oh, maybe these are conditions right for some really interesting microbes," or was it by accident? How did people come to know thermophiles in Yellowstone?
Yeah, another really great question. And I love history. I like microbiology history. I love the history of scientific exploration in Yellowstone. Geologists back in the 1890s were the first to describe what would become thermophilic microbial species. Now, they didn't know that what they were looking at were bacteria or archaea or even algae. They called them like thermophilic plants. They thought they were plants. And so you can go to these hot springs in Yellowstone. I said you can't see microbes with your naked eye, but they can form macro communities. And you can see these floating in the outflow channels of hot springs all over the place in the park. And so you can see them, and they do kind of look like plant matter, if you will. So that was in the 1890s, but it wasn't until the early 1960s when a microbiologist from Indiana University was traveling across the Western United States with his family, a guy named Thomas Brock, and they stopped through Yellowstone on their way to California, I believe, and he was surprised to notice what looked like microbial biomass in the outflow channels of some of the hot springs in Yellowstone. And on his return trip, they actually sampled some of those and put them under a microscope, and sure enough, they were bacteria. At that time, they didn't know archaea existed, but there was archaea there as well. So, 65 years ago, something like that, was the first time that somebody really pinned microbes living in hot springs in Yellowstone. And it's been a fun scientific journey since then for our entire community.
And just on a family vacation. I love that. um, do different types of thermophiles live in different types of thermal features, like, say, hot springs versus mud pots versus maybe geysers and everything in between?
Yeah. Um, there's really four different types of hot springs, or thermal features, I should say. There's hot springs, there's geysers, which are simply hot springs that periodically erupt. There's fumaroles, which are just essentially steam or vapor vents. And then there's mud pots, and mud pots are kind of similar to fumaroles in the sense that there's just really very little water, right? And so you're really talking about the amount of water in the system that differentiates those different types of thermal features. Um, and so, yes, the answer to your question is yes. Different microbes live in different hot springs, fumaroles, mud pots, geysers. Um, and what's really peculiar about Yellowstone is you can kind of break the different hydrothermal systems up as either being acidic and smelling like rotten eggs and having a bunch of yellow sulfur in them, or being alkaline.
Mhm.
And so geysers tend to be alkaline. If you see a hot spring that is bright blue and clear, those tend to be your alkaline circumneutral hot springs. Mud pots and fumaroles tend to be acidic. And that's because they're primarily volcanic gases enriched with sulfur. When you oxidize sulfur, you make sulfuric acid, which is acidic. And so you kind of differentiate those two different hot spring types, and the biology is dramatically different between those two hot spring types. So again, geysers and clear blue hot springs tend to be alkaline or circumneutral, and then fumaroles, these gas vents and mud pots, uh, tend to be acidic.
How do you sample from a geyser? I have to, like, wonder about that. How do you know just, like, wait, time it, wait till it erupts, hop in there?
Yeah, well, I tell you what, we had to figure this out several years ago, and so this may surprise people, but um, nobody knew whether or not microbial life lived in geysers. And part of the reason for that is that, well, it's exactly what you just asked. How do you safely sample from a geyser? Because that water is boiling when it's coming out of most of those geysers, and you do not want boiling water to hit your skin. It is not a good place to be. So we convinced the park that this was an interesting question, an interesting enough question to ask, and they permitted us to sample Old Faithful. Now, Old Faithful is the reason they let us do it was one, you're close enough to infrastructure so that if something did happen, you could get help there fast enough. Two, it's faithful. Its eruptions are faithful, right? So we could time it. We could predict. You know when it last went off, and you know when it's going to go off again, and so you can safely give yourself a window of say 20 minutes in the middle to set up your sampling materials, and then get out of there when the eruption happens. And so when we sampled Old Faithful, what we did is we sterilized just simply a bin, like a bin that you would use to store your clothes. We sterilized it, we weighted it down, and we just stuck it up on the cone to collect geysed water. Um, and then got away from the cone to a safe location for when the geyser went off. But it's one of the critical questions, and if you've been to Yellowstone, and I hope everybody that's hearing this podcast has, or at least plans to go, you'll notice all of the signage all around the park telling you how dangerous it is to walk off boardwalk or to walk in thermal areas. It's very dangerous, and you got to take every precaution. And so that's why the park does not allow you to do that. It's illegal to do that unless you get permission from the park itself.
Absolutely, to do some science. Um, can you tell me a bit about the role that thermophiles play in the Greater Yellowstone ecosystem? Are they a part of the food web, or, do they I don't think they contribute to any decomposition, but yeah, what is their role in this ecosystem?
Yeah, well, it's this is gonna blow people's minds. At least I hope it does. Okay, and so you may not think that a thermophile has any role in the ecosystem. They live in these island-like environments, these acidic or alkaline high temperature environments, the geyser basins, if you will. Well, what if I told you that the generation of acid in the park, so if you go to Norris Geyser Basin and you look around, Norris Geyser Basin's been there for about a quarter million years. You'll notice that you're in a depression. And most acidic geyser basins are in a depression because acid eats rock. So over the last 250,000 years, Norris Geyser Basin has been actively eating rock. But what if I told you that the generation of acid in these hot spring environments is due to the microbes, the thermophilic microbes that live there? They are the ones that catalyze the oxidation of sulfur compounds to generate sulfuric acid, which is the source of the acidity in the park. And so, anytime that you're in a geyser basin, in particular places on the northern edge of the park and on the eastern edge of the park where most of the acidic geyser basins are, you can ask yourself, well, without those microbes, without those thermophilic sulfur oxidizers, these geyser basins would look dramatically different. They wouldn't look like the moon-like or alien-like landscapes that they do look like. So that's one example of how thermophilic microbes um, impact the ecology of Yellowstone, if you will. Another way that they do so is they transform a lot of elements into different forms that are either less toxic or more toxic. So I'll give you an example of mercury. So microbes, volcanic systems, just in general, it doesn't matter where you're at, tend to have a lot of mercury associated with them. Okay, a lot of mercury mines, places where they used to mine a lot of mercury, are in old volcanic terrains. Okay, so Yellowstone's no different in that. There's a lot of mercury in the Yellowstone thermal waters. Well, microbes in these hot springs actually transform mercury into methylmercury. They add a methyl group, a CH3 group, onto that mercury. And methylmercury is a potent neurotoxin. It is extremely toxic to humans, to other animals as well. And that methylmercury, once it's made, biomagnifies through food chains. And so if you catch a fish, say in the Firehole River, there's a decent chance that that fish is going to have a lot of methylmercury associated with it because the waters that form the firehole are thermal waters, right? Go to Excelsior Geyser, go to the Middle Geyser Basin, and watch the amount of water flowing out of that hot spring, those hot springs in the Middle Geyser Basin into the firehole. It's extraordinary. Well, all of that water is going to have all of that mercury, all of that arsenic, so on and so forth, um, that came into the volcanic system and is now draining into all the waters and the watersheds that then leave the park. So varied influences on the ecology of Yellowstone, but they certainly do. The thermophiles certainly do have an impact on the ecology of Yellowstone.
Yes, my mind is very blown. Could you just tell me a little bit more about how thermophiles can turn mercury into methylmercury? I don't know, my brain just can't even wrap around that. What is the chemical processes there?
Well, it's there's a set of enzymes, and so you might ask, maybe the question to ask, I should have framed this a little differently, but mercury is toxic in its inorganic form as Hg2. It's toxic. It's also toxic as methylmercury. Okay, but for these microbes, the inorganic form, the Hg2 form, is more toxic to them than the methylmercury form. And so they methylate mercury. It's an enzymatic process mediated by proteins in those cells. And what they're doing is they themselves are detoxifying that mercury. For they just put a methyl group on it. It makes it less reactive in their cells. Now, it doesn't help us any. It doesn't help eukaryotes any because it's more toxic to them. But that's why they do it, is to detoxify their local environment. So essentially, it's, not to get into the biochemistry, but it's a coronoid protein that these cells encode, and they detoxify their local environment of mercury by putting a methyl group on it. And of course, since these hot springs are not hydrologically disconnected from the watershed, that methyl mercury is going to go somewhere. And so those microbial mats that you see in some of these hot springs, well, those are a food source for other organisms. And so you'll see flies, you'll see larvae, so on and so forth, on those mats. And guess what they're doing? Well, they're eating those mats. And that's how it makes its way into the local food chain. Well, what do fish eat? Fish like to eat insects. And so there you go. That's how this whole, what you would call a biomagnification, how that occurs. And it's no different. The process is no different than the way that tuna fish or other predatory fish become enriched in methyl mercury. And so if you're a pregnant woman and you're hoping to maintain the health of your soon to be baby, you're advised not to eat a lot of tuna fish because there's a lot of methyl mercury. Methyl mercury is a neurotoxin, so you don't want to eat too much of that anyway. Well, the way that those fish got enriched in methyl mercury is because they have to eat about 10 times their body weight to produce that amount of biomass. And so by doing that, they have a potential to biomagnify at each trophic level tenfold the amount of mercury in their tissue, and so on and so forth. And the same process is happening in the hot springs of Yellowstone.
That is so fascinating. Thanks for diving a little more into the details on that. Um, how were you drawn to the field of microbiology and eventually thermophiles, and then maybe eventually to Yellowstone in particular?
Yeah, when I was, people always say that you can't remember anything before you were, say, 5 years old. Well, that's not true. That's some of the solidest memories seared into my mind are from before I was 5 years old. And one of those memories was going to Yellowstone with my family. I grew up in Iowa, and so we would go west on family vacations, camping vacations, and we went and stayed for a couple days in Yellowstone, and it was just blown away. Um, and this is kind of a long kind of story here, but I'll try to keep it as simple as I can. I also had an interest in minerals growing up. Love to collect rocks. Love to collect minerals, so on and so forth. When I was in college, I went to college. I wanted to be an orthopedic surgeon. Um, Long story short, I went to a field biology station in Colorado and developed a fascination with ecology and specifically microbiology, and went back to my university and switched my major from wanting to be an orthopedic surgeon and a pre-med to a microbiology major. Um, and so I had these separate interests: microbiology, minerals, and in the back of my mind, I couldn't forget Yellowstone. And then a professor from Montana State University by the name of William Costerton, who was the director of the Center for Biofilm Engineering here, a former priest, I should mention. So, a very eloquent speaker, a very convincing speaker, came to my university back in Iowa and gave a seminar and talked a little bit about these microorganisms that formed biofilms on minerals and rocks in hot springs in Yellowstone. And I thought to myself, well, there's no better thing to do with your life than to study and dedicate yourself to study microbes that live off of minerals in hot springs in Yellowstone. And so that's maybe a longer answer to what should have been a simple question, but that's how I got to where I'm at. And Couldn't be happier.
Yeah, I just love the effect that Yellowstone has on, I mean, myself and so many people I've had on this podcast where it's a visit at a young age or even sometimes a little older, and I think we can all just agree that this place just hooks you and calls you back. And I had a similar experience that came out here, visited, and just always felt like I needed to come back, and here we are. It's awesome. Could you tell me a little more about what an average day in the field could look like if you're in Yellowstone collecting samples? You talked a little bit about your experience with Old Faithful, but is it mostly sample collection and then you're taking it back to your lab? Is there any assessment in the field? Yeah, what does the average day look like?
Yeah, and so it depends on what question we're asking, and it depends on what location in the park we're going to go to. But just on in general, one of the benefits that we have in being at Montana State University, as the crow flies, I can be at my field site in 2 hours, and and so I can do things that other people can't necessarily do at other universities. And so we have that benefit. So a lot of the work that we do is in the form of day trips. So we'll get up early. I actually live in Livingston, so usually somebody will drive over I-90 over the pass and pick me up, and we'll drive down. We'll try to get into the park before the big slug of tourists come in. Get to our field site. Um, and I should mention, most of the work that we do, we can't do in the public view. So we have to travel into the backcountry to do this. So there will be a little bit of hiking. And we'll go set up shop next to one of the thermal features that we want to study. And typically what we do is we characterize the chemistry and the temperature, so on and so forth. We'll make a bunch of chemical measurements on the hot spring before we do anything else. A lot of times we're actually trying to quantify microbial activities. So the way that we know, for example, that biology is driving the oxidation of sulfur compounds is because we can go to the field and we can measure those activities. And we can set up different experimental controls to attribute those processes to the microbes or to not. So we'll run some activity assays, for example, or we'll grab material and flash freeze it on site for transport back to Montana State for, say, DNA or RNA based analyses. Probably what my lab is best known for, though, is collecting samples to then bring back to the lab to grow those organisms in the lab. And so we are trying to domesticate, if you will, the microbes in our ovens that we keep in the lab. And then once we have those organisms in the lab, we can characterize them and figure out what they can and cannot do. And again, to kind of go back to one of the original